So today we're going to be looking at the reproductive system, and we're going to spend most of the time today looking at the male reproductive system. But before we do, I want to talk about it in just a little bit in general. So if you look at humans, we have what's called a life cycle. And so the human life cycle, when you look at it, so it starts out as the zygote. Hopefully everybody in here knows what a zygote is, and then we're going to grow and then develop and so on. Well, when we talk about reproduction, humans actually have two different types of reproduction. One type that we have is called cellular reproduction. And cellular reproduction is also said to be asexual. It does not require sexes or anything like that. And basically what it is mitosis. And so one cell is going to become 2, 2 is going to become 4, and 8, and 16, and so on. And we use this cellular or asexual reproduction for growth. We use it to repair damaged tissues and so on. And so everybody in here started out as something that looks like this, but you go from one cell to trillions of cells, and all of those cells are basically exactly alike in terms of their genetic information. There's no difference. And that's what you get with mitosis. And there are some animals that only do asexual reproduction. But humans aren't like that. Humans have sexual reproduction. But sexual reproduction has a problem. So if you look at our body cells are 2N. What that means is that our chromosomes come in pairs. And we have 23 pairs of chromosomes. So if we took just a plain old body cell from a male, and we took a plain old body cell from a female, if we were to combine those two together, this is 23 pairs, and this is also 23 pairs. That new individual, that new organism, would have 46 pairs of chromosomes. In other words, they'd have twice as much DNA as their parents. And then if it happened again with a pair of these, you'd have 92, and so on. And so what would happen is with every generation, you would increase the amount of DNA in the cells, and eventually there wouldn't be room for anything else except DNA. So we have to do something to prevent that from happening. And so animals that use sexual reproduction, they make special cells, and the special cells are called gametes. And so gametes, when you look at a gamete, here they are, they're special cells. They're not diploid. They're not 2N. They're 1N cells. And because they're 1N, they only have 23. Not 23 pairs, but 23 total chromosomes. So that means there's 23 in here, and that means there's 23 in here. And when you combine 23 plus 23, we get back to this 2N or 46 cell. Well, we have to use a different process to do this. So for cellular reproduction, for asexual reproduction, we're going to use a process called mitosis. But for sexual reproduction, we have to use a different process. And so that process is called meiosis. And so we're going to take a few minutes and look at mitosis and meiosis. So here's about as simple as you can possibly get. So remember, mitosis is asexual. What we're going to do is we're going to take a plain old body cell, which is 2N. Remember, that's also called diploid. And we're going to go through this process, and what we're going to wind up with are two cells that are exactly like each other and exactly like the original, except they're smaller. But they're also 2N, they're diploid. But in its simplest form, meiosis takes a 2N cell, and we're going to go through a process twice, and what we're going to get is not 2 cells, we're going to get 4 cells. And those 4 cells are not like each other, and they're also not like the original. and they're not diploid. These are haploid cells. And so let's see how all of this works. So let's look at mitosis first. We'll come back to this slide. Diploid, haploid. So diploid, 2 chromosomes. Haploid, 1 chromosome. So let's look at mitosis and meiosis. Well, a good way to think about chromosomes is to think about like shoes. Look, shoes come in pairs. But even though these are a pair of shoes, they're not exactly alike. Look, that's a right shoe and that's a left shoe. And you really can't wear the left shoe on the right foot or the right shoe on the left foot. They're not exactly alike, but they're close. But look, there's 3 pairs of shoes here, 3 pairs. And so this would be an example of this 2N because they're in pairs. And chromosomes are exactly the same way. Look, there again, there are 3 pairs here. And because they're pairs, 1 pair, 2 pair, 3 pair, they are 2N cells. And so that's what we're all going to start with. And this is what all of our body cells are like. They're 2N. They're diploid. Well, when we go into mitosis, what's going to happen is, well, first of all, we're going to copy the DNA. We're going to duplicate it. So when we duplicate the DNA, we're going to get exact copies of this, an exact copy of this, and so on. And so now we're going to have 2 copies of each pair. So if this were a pair of shoes, and that was the left one, and this one was the right one, well, now we're going to duplicate that. So when we look down here, that's 2 left shoes, and that's 2 right shoes. And these 2 are exactly alike, and these 2 are exactly alike, but this pair, this is not the same as this. Remember, we have left shoes and right shoes are not exactly the same. But now look, we have 2 times the genetic information that we did up here. And so the next thing that's going to happen is we're going to separate these identical copies. We're going to split them up. And we split up the identical copies into separate cells. And so each new cell is going to be back to having pairs again. Again, there will be pairs. And those pairs are going to be exactly like the original pair. So there's no variation here. And remember, some animals, some organisms only do this kind of reproduction. Well, there's some advantages to it. It's fast. You don't need to find a mate, things like that. But there's a big disadvantage to this. And that is if something, some virus, some bacterium or something can kill this cell, well, these are exactly like it. So not only can they kill this one, they can also kill these as well. And so you can wipe out the whole population pretty quickly and pretty easily. Meiosis is different. And so what we're going to do with meiosis is we're sort of going to do the process twice. But we're going to start out with the exact same cell we had before. And look, there's pairs of chromosomes, 3 pairs. And we're going to duplicate them again. So there's our duplicates, there's our duplicates, there's our duplicates. But then something different is going to happen. What's going to happen is this pair and this pair, where there are 2 identical copies, are all going to hook up. So look, our pair has 2 identical copies here. They're all 4 going to hook up. And the word for 4 is tetra. So these are called tetrads. That did not happen in mitosis. So look, we have 4, 4, 4. We have these tetrads. And then something completely different is going to happen. What we're going to do is we're going to separate the pairs. Remember, these are two identical copies, and these are two identical copies. If we use our shoe analogy again, these are both right shoes, and these are both left shoes. Well, we're going to separate the pairs. We're going to put all the right shoes on one side and all the left shoes on the other side. They're no longer pairs. So this is a 2N cell because it has pairs. This is a 2N cell because it has pairs, but this doesn't have pairs anymore. This is a 1N cell. But it still has two identical copies, two left shoes, two left shoes, two left shoes, two identical copies of these individual chromosomes. And so we're going to go through the process again. And this time what we're going to do is we're going to separate the identical copies. And it's going to go this way, and that one's going to go this way, and so on. And what we're going to wind up with, instead of 2 cells, we're going to wind up with 4 cells. And the 4 cells are not alike, and they're not like the original. If we go back to that other slide, you can see how they're not alike. Look, those are all red. Those are all orange. That's all blue, dark blue, light blue. But what happens when we do this whole process is we wind up with 4 cells that are not like each other anymore because everything is all mixed up. And so if something can kill this or even this, it may not be able to kill all of these. And so when we do sexual reproduction, it has some disadvantages and advantages too. One disadvantage is you have to find a mate, and that's not always easy. But a big advantage is you increase the amount of variation in the population. And the more variation there is in the population, the less likely that you're going to be able to wipe out the whole thing. There may only be a few that survive, but those few that survive can carry on and replenish the population. So, again, big difference between body cells. They're diploid. They're 2N. And these special cells, gametes, they are haploid. They're 1N. Anybody have any questions? So to summarize that, with mitosis, we're going to take one cell, it's going to divide, and we're going to get 2 identical cells. They're exactly alike, and they're exactly like the original cell. So there's no increase in variation. There's no increase in variation. And so if you take any cell from anywhere in your body and you look at any other cell, hand, foot, wherever, if I take it from here and then I come over here and I take them here, they're going to be identical in terms of DNA. There's no increase in variation. If we summarize meiosis, though, we're going to take one cell. We're going to go through a process twice, and we're going to get 4 cells that are not alike and not like the original. And we're going to increase the variation. And so what's going to happen is we're going to have no 2 cells that are alike. They're all unique. Every single one of them is unique. Any questions? So look, there's our original 4 cells, our 4 chromosomes. We go through the process, we multiply, and this is what we're going to get. 4 cells that are not alike, and they're not going to be like our original either. Any questions? So, if we look at mitosis, that happens everywhere in your body. Basically all cells, all organs, it's everywhere. But meiosis only happens in very, very specific places. Only in one place in your whole body. And that place is in the sex organs. That's the only place where meiosis happens, nowhere else. There's another difference in meiosis, though, depending on if you're a male or a female. Now, we're not going to talk about females today, but we'll be a little bit later on. But remember, we're going to start one cell, and we're going to go through this process twice, and we're going to wind up with these unique cells. Well, in males, when we do this, we're going to get 4 unique gametes, 4 of them. But females, they go through the whole process twice, just like here. But what happens is you wind up with 1 big cell and 3 tiny little cells. And these tiny little cells are called polar bodies, and they die. So that 1 big cell is the only cell we're going to wind up with. So we're not going to get 4. We're going to get 1 functional cell and 3 non-functional cells. So it's still 4, but they're different. Does it make sense to everyone? Any questions? Okay, so we're going to look at males. It's a little easier to look at males than females because it's a little simpler. But both males and females have primary sex organs, and the primary sex organs are called gonads. And remember, this is the only place where meiosis happens. And gonads in the males are called testes. Gonads in the females are called ovaries. And this is the only place where we're going to do gamete production. These gonads, though, also are endocrine organs because they produce hormones. And so we'll see these hormones, which are called the sex hormones. But the gametes production is not enough. Humans have internal fertilization. Not all animals do. Some animals just release their gametes into the environment and hope they find each other. But humans aren't like that. We have internal fertilization. Well, because it's internal, it's a lot more complicated. And we're going to have a lot more structures to aid it. So there are accessory structures, accessory organs. And these are things like ducts and glands and external genitalia and so on. So we have the primary sex organs, and then we have the accessory sex organs. So let's look at the male. So here's a sagittal section through the male pelvis. And before we talk about the whole structures and everything, I kind of want to just do a little tour through here and just name the organs. So here's our primary sex organ right here. That's testis. There's 2 of these. And they're located in a skin-covered pouch called the scrotum. Well, this is where the gametes are made, and male gametes are called sperm. So this is where the sperm are made. And then the sperm leave this and they enter this structure here. It's called the epididymis. It's sort of comma-shaped. And during this time that they're in the epididymis, they're going to mature. And by the time they travel all the way through here, they can fertilize an egg and they can swim. And then they just sort of sit here and they wait. And they wait for the male to become sexually excited. And then they enter this tube. There are 2 of these as well. And then this enters or re-enters the abdominal cavity. has little muscles all along it, and those muscles push the sperm up this tube. So they don't have to swim, they're pushed along. And as the male becomes more and more sexually excited, they're pushed further and further up the tube until they get to this little area here. That is called the ampulla. It's the last part of this tube, which is the ductus deferens. And they basically sit there and wait. And they wait until the male becomes sexually excited enough for ejaculation to occur. And then those same muscles that moved them through here contract and forcefully push them this way into this tube, and that's called the ejaculatory duct. Right next to this ampulla is a pair of sex accessory glands. These are called the seminal vesicles, and they make a fluid that gets added to the sperm. So as the sperm move by, there's also muscles in here, and the muscles contract and force the fluid. So now there's a mixture right here. And then we enter another gland, and this one there's only one of. It's called the prostate gland. It's shaped like a donut, and this tube goes right through the middle of it. Well, as it goes through there, this also contracts and forces more fluid. And then it continues through, and then this is the penis. And then it comes out to fertilize the egg. There's also one other pair of glands. They're these little bitty glands right here. They're called bulbourethral glands. And those little glands produce a fluid that enters this tube before ejaculation. And some of it sometimes comes out and forms this sort of slicky layer that aids in lubrication. But those are the organs. So testis, epididymis, ductus deferens, seminal vesicles, prostate, bulbourethral glands, and penis. And then that's what we're going to look at. If you dissect it out, it looks like this. Same thing. There's the testes, there's the epididymis, there's the ductus deferens. Remember, there are two each of these. You come around, there's the bladder, but on the back side of the bladder is the seminal vesicles. There are two of those. There's the prostate, and then little bulbourethral glands are here. There they are right here, and then we have the penis. Everybody got all that? Anybody have any questions? Now we're going to look at each one of these individually, but right now you should be able to kind of go through and name the organs. Here's a real human one that's been dissected out. You should see the same things. So there's the testis, the epididymis, the ductus deferens. There's our ampulla, the bladder. And then here's the seminal vesicles. There's the prostate. There's the little bulbourethral glands. And then here's the penis. Okay, so let's look at the organs. And the first organ we're going to look at is actually a pouch. It's a skin-covered pouch that contains the testes. It's called the scrotum. So if you look at a fetus, and here's a fetus that's 2 months old, the scrotum is empty. It doesn't have anything in it. That's because the testes don't start there. The testes start at the same place where the ovaries do in the female. It's in the abdominal cavity. But look what happens. As this fetus develops, they move. And they move in this direction until right around the time of birth, they enter the scrotum. Well, you might wonder why. There has to be a pretty good reason, because think about it. Depending on how you look at it, the testis might be the most important organ in that male's entire body, because that's where the next generation is going to come from. And if there are no more testes, there are no more next generation. So why would you put such an important organ out here in this very unprotected place? There has to be a good reason. Think about it. Your heart. Your heart is in your chest cavity. It's surrounded by the ribs and so on. Your brain and your spinal cord are encased in bony, hollow cavities. There's bone that surrounds all that, but there's nothing like that here. And there's a really good reason for it. So there's the testis. We're going to watch it as you develop, and eventually it's going to enter the scrotum. So 2 months, 3 months, 4 months. Getting close to the end of pregnancy, they leave the abdominal cavity and they pass through a hole in the abdominal wall. That hole is called the inguinal canal. And look what else happens. As they move, look, there's blood vessels attached. There's nerves attached. The vas deferens, the ductus deferens is attached. And so all of that is going to get pulled along with it. And then right around the time of birth, it enters this scrotum. Some babies are born before it happens. This happens before birth. Sometimes it happens after birth. But there's the testis, there's the epididymis, and then there's the blood vessels and the ductus deferens. Again, there has to be a good reason for this, and there is. And the reason is temperature. If you look at core body temperature, it's hot. Our normal body temperature, remember, is about 98.6 or whatever, something like that. But the core body temperature is actually a little bit higher than that. You cannot make sperm at core body temperature. They either come out dead or they come out abnormal. You just can't make them at core body temperature. And so you have to somehow make them cooler. Well, the only way to do that is to remove them from the abdominal cavity. You need a temperature about 3 degrees Celsius less than core body temperature. And so they enter this pouch, the scrotum. Well, when we look at the scrotum, it's a fairly complicated organ. It's not just skin. It is skin on the outside, but there's a lot more to it. There are 2 sets of muscles that are part of it. 2 sets of muscles. One is skeletal muscle and one is smooth muscle. And if you look at this picture, you can see the skeletal muscle right here. It's part of this internal oblique abdominal muscle. So fibers from that muscle move down along with the testis. This is now called the cremaster muscle. So the cremaster is skeletal muscle. Remember, skeletal is voluntary. So males have a little bit of voluntary control over their scrotum. Not much, but some. There's another muscle too, and that's this muscle right here. And this is smooth muscle, and it's embedded in the skin of the scrotum. And so it gives, when it contracts, it gives the skin a puckered appearance. But this is smooth muscle. It's called the dartos. So the dartos muscle is this smooth muscle that wrinkles the skin of the scrotum. And then there's one other thing to help maintain this 3 degrees Celsius lower than normal body temperature lower. And that is blood vessels. Remember, blood vessels come down with the testis. Well, if we look at the arteries, they're very, very straight. They're just straight, plain old, normal-looking arteries. They're either called the gonadal arteries or the testicular arteries. but they're just straight arteries. And the blood that comes through an artery, this comes directly off the aorta. That blood is hot. It's core body temperature. And that hot blood, as it come down here, would heat up this, and it would be too hot. So something happens to cool the blood before it gets here. And that is, there's a network of veins. All these blue things that you see here are veins. They're not normal veins. Remember, veins are usually straight and they run right alongside the artery. They run just side by side. But look at these veins. They form this weird little network wrapped around the arteries. And what happens is the heat from the artery moves into the veins. And so the blood in the artery is less hot as it gets down here, and less hot and less hot, until when it gets here, it's not hot anymore. It's a little bit cooler. If you take a thermal camera and take a picture of a male scrotum and testes, up here, it looks like this. It's dark, dark red because this is very hot. Hot blood is coming out of here. But remember, what's going to happen is that heat is going to leave the artery and travel to the veins. And so a little bit lower, it gets cooler. So now it's a little cooler as you get them further down, but more heat is going to leave. And so as you move down a little more, it's even cooler. And if you move down even more, it's even cooler. So by the time you actually get to the testis, the temperature has dropped dramatically. Well, that network of veins is called the pampiniform plexus. The pampiniform plexus. So the pampiniform plexus is this network of blood vessels, these veins, and they also help to lower the temperature. So these three things then have to do with temperature. Well, not only do we want the temperature to stay about 3 degrees less than core body temperature, we want it to keep close to that as possible. So we want to stay as close to 3 degrees less as we can get. Well, sometimes it might be cold out here. And if it's cold out here, then this is going to be cold as well. Well, cold also interferes with sperm production. And so we need some way to heat the temperature back up. And so what happens here is these muscles, the cremaster and the dartos, contract, and they lift the testes up. They pull the testes up closer to the body wall. And they're closer to the body wall, and it's warm up here. But they can also relax. So if they relax, the testes are going to move further away from the body wall, out here into the air where it's cooler. And so depending on the ambient temperature, the temperature around, this is constantly happening. They're going up and they're coming down, depending on the temperature. Any questions about those three things? So that's pretty much the scrotum. Now don't forget the scrotum is covered in skin. It's also divided into 2 compartments. There's a wall here in the middle. But it's basically just a skin-covered pouch. And it contains the testes, the epididymis, the ductus deferens, these blood vessels, these muscles, all those things that came down. And if you lump all those things that came down together, It's called the spermatic cord. So the spermatic cord is what passes through that inguinal canal. It's wrapped in connective tissue, dense, irregular connective tissue. Make sense? Any questions? Okay, there's one other thing that gets pulled down. Let's go back to our other picture here. Remember, if we look in the abdominal cavity, there's a membrane right here. Double layered membrane. Remember, it's called the peritoneum. Well, when this testis gets pushed down, it doesn't go through the peritoneum. What it does, it drags it with it. So there's peritoneum down here. So if we look at it, it looks like this. It comes this way, and then it goes down in here like that. And then back up, and then over here, and then it goes down in here like that. And then back up here. So there's another layer in the scrotum, and it's the peritoneum. And you can see it right here. It's very thin, it's simple squamous epithelium. Remember, the serous membranes have two layers. There's a visceral layer and a parietal layer, and there they are right there. This peritoneum. But it has a different name down here. It's called the tunica vaginalis. But you can see the two layers right here, one touching the organ, one not touching the organ. Again, that is the peritoneum, but it's called the tunica vaginalis. So you might think it's a little weird. Males have a tunica vaginalis, but females don't, because females don't have this whole process that happens. Any questions? Okay, here's another view of that same thing. Here's the inguinal canal right here. You can see what passes through it. Remember, it's wrapped in connective tissue, and then here down here you can see all the things that are inside the scrotum. Okay, well, the main thing that's in the scrotum are the testes. So let's look at the testes. So first of all, when you look at the testes, remember we've got this tunica vaginalis, this peritoneum that came down with it. So that is simple squamous epithelium. It's very, very thin. You can even see through it. But then the testis itself also has a wall around it. And it's made out of fibrous connective tissue. It's dense, irregular connective tissue. Remember, dense, irregular, when you look at it, it's white. And so the testis has a thick white coat around it. And there it is right there, that thick white coat. It's called the tunica albuginea. Alba means white. So it's a thick white coat of this irregular connective tissue. But not only does it surround the testis, it also goes inside the testis. So here's that tunica albuginea right here. But look, it folds in right here, and it folds in right there, and it folds in. And it forms these sort of walls inside the testis. The word for wall or separation is septum. And what it does is it creates little compartments right here. So when you look, there's several of these little compartments. Well, each compartment is called a lobule. We can also in this picture see the tunica vaginalis. That's what this is right here. That's our tunica vaginalis. Again, that's the peritoneum. Don't forget there's a little space in between these two layers. There's a little tiny cavity. But those are the walls. And so we create these lobules. And then if you look inside each lobule, you can see them here. There's a lobule, and there's one, and there's one, and there's one. Here, over here, same thing. This is a real one. There's our little septa. And you can see the lobule in here and the lobule in there. Well, inside each lobule is something called a seminiferous tubule. So seminiferous tubules, when you look at them, they're long little tubes. Look, they're blind on this end. They're not open, but they are open on that end. But it's a long, all twisted up little tiny tube. And it's twisted like crazy. So if I were to make a cut across this tube, when I look at the cut, sometimes the cut's going to be round, sometimes it's going to be oval, sometimes it's going to be long. It depends on how it's been cut. And that's exactly what we see when we look right here. Look, some of these are round, some of them are oval, some are cut long ways. So we have transverse sections, remember that from A&P 1? We have oblique sections, and we have longitudinal sections, all here in this one picture. Again, it's a tube. These are tubes, and tubes are hollow. So look in the middle of it, and you can see it here, a little bit lighter spot in the middle. There's a hollow space, and the hollow space is called the lumen. Well, if we look at the cross section of it, there's the lumen right there. Now, in this particular view, all I see are transverse sections because they're all round. But there's one tube that's been cut. There's another tube that's been cut. Another one, and so on. Well, if we look at this tube, the seminiferous tubule, this is where the sperm are made. This is where sperm get made. And as the sperm get made, they go through stages. So here at the outside edge of the tube, that's the least developed cells. But what's going to happen is we move this way, we're going to go through meiosis. As we go this way, we're going to go through meiosis. And so as you move further and further away from the outside toward the center, you get more and more developed cells. So you can even see meiosis going on in here. Look, you can see the chromosomes folding up, and you can see different stages of that. And so when we look out here, here's our plano-normal body cells. Remember, those are diploid. They're two-end cells. But as we move this way, we're going to go through meiosis, and they're going to divide. And then we're going to keep going, and they're going to divide again, so that we wind up with 4 cells. And those 4 cells, remember, are not going to be diploid anymore. They're haploid. And that's what happens. The average human male makes about 100 million sperm every 24 hours. So this is a process that's happening very, very quickly and in great number. But look, we have tons of these seminiferous tubules. Look at how many there are. And every single one of them is making those sperm. Another thing you can see in the seminiferous tubules is there's a different type of cell in between them. So these cells are doing something completely different. These are called interstitial cells. And interstitial cells don't make sperm, they make hormones. So these cells are endocrine cells. And the hormone that they mostly make is testosterone. So the testis, not only does it make sperm, it's also responsible for making testosterone. But it happens here and here and over here. It happens between these seminiferous tubules. We go back to the other picture, we can see them here, right here. These are those interstitial cells. Happens between. Any questions? Okay, so we're going to make sperm. And remember, we're going to make about 100 million every 24 hours. Well, they've got to go somewhere. They've got to get out. And so what happens is those seminiferous tubules, the ends of them converge, and they form these little straight tubes called tubulus rectus. So look, there's the ends of the tubes right there. That's the tubulus rectus right there and right there. Little straight tubes. They're not very long and they're not very big. And so then they're going to go into this structure. And this structure right here is a little network looking thing. It's called the reedy testis. And from there, we're going to enter a completely different organ. And this organ is this one. This is the epididymis, and it's not part of the testis. It looks like a comma on the side of the testis, and it's this long, long, long tube. So let's look at this epididymis. So, again, it looks like this. It has a head, it has a body, and it has a tail. What happens is the sperm enter the head, and they travel through here. And as they travel through, what happens is they mature. So sperm that can't even swim enter the epididymis. And they pass through this long tube and they develop the ability to swim. They also develop the ability to fertilize an egg. And they're going to stay in the epididymis until right prior to ejaculation. And the epididymis is going to push the sperm into the ductus deferens. So in the walls of this epididymis, there's smooth muscle. It also has tissue on the inside. So here's our epididymis right here, long tube. And if we do a section across it and look at a hollow space, there's the lumen right there. And look, the lumen is full of sperm. You can see millions and millions and millions of sperm in there. Well, here's the wall of the epididymis. There's smooth muscle right there. And if you look right here, this is pseudostratified columnar epithelium. Remember, pseudostratified has cilia. You can see the cilia right here. And so what this pseudostratified does is it secretes stuff that help the sperm mature. It also absorbs the waste products from the sperm. And so it helps maintain or keep the sperm healthy until they get ready to be ejaculated. But they can't live in here forever. And so sperm will also die in here. And when they die, this epididymis will reabsorb them and get rid of them. So here's our epididymis. Again, it's sort of a comma-shaped organ. And there's the head, there's the body, down here is the tail. And so down here at the tail is where mature sperm that can swim, that can fertilize an egg, that's where they're stored. This is also the part where if they die, they're reabsorbed. But look, here's our seminiferous tubules. There's our little straight tubules right there, and the reedy testis. And then there's our epididymis. Any questions about the epididymis? Okay, well, let's look at the penis. So the penis is the organ of intromission or copulation. Remember, humans have internal fertilization. And so in order to accomplish internal fertilization, there has to be this organ that penetrates the female reproductive tract. Well, the only way that can happen is if it's a rigid organ. Well, some animals actually have bones in their penis, and that's what makes it rigid. But humans do not have bones. What we have instead is what's called vascular tissue or vascular spaces. And this vascular tissue fills up with blood, and so it becomes turgid. And once it becomes turgid, then it becomes stiff and elongated. So humans don't depend on this. What we depend on is almost like hydraulics. But it's covered in skin, and if you look at it, the skin extends across the end of the penis and forms what's called a prepuce or foreskin. So it's this sort of cuff of skin, and that cuff of skin is retractable. So when the male becomes sexually excited and the penis becomes enlarged and stiff and turgid, mostly that is retracted. Sometimes the skin is removed, and that's called circumcision. Sometimes it happens right after birth for religious reasons or maybe for aesthetic reasons. It sometimes has to happen as an adult. But it's not a very normal thing to do. No other animals do that. But it's a choice, just like any other kind of body modification. But anyway, if you look at the penis, look, it looks like this. And it's got 3 parts to it. It has a root. A root you can't see, it's hidden. Then it has the shaft, and then it has the glands. So we've got a root, a shaft, glands. So let's look at it in a little more detail. So remember, we've got these vascular spaces, and they're enclosed. They're chambers. And if you look, you can easily see there's 3 chambers. Well, 2 of these are enclosed in connective tissue, just like the testis is. And in fact, it has the exact same name. It's white, so it's called the tunica albuginea. Remember, this is dense, irregular connective tissue. So what happens is this penis fills up with blood, it pushes against this connective tissue. And that's what creates the increase in pressure and the turgidity and rigidity, and so on. There's also a third one that's not enclosed in this connective tissue. And this third one has in the very center of it the urethra. So the urethra runs right through there. So it's a good thing that it's not enclosed in this connective tissue, because the urethra would be forced to collapse and then sperm would not be able to get out. But in the center of this is an artery. So look, there's an artery right there, an artery. There it is right here. There's the artery. It's this artery inside the chambers. Notice there's nothing like that down here. We have smaller tissue spaces, but no main artery. So let's look at these chambers. So again, there's 3 of them. So there are 2 which are called the corpora cavernosa. And here are the corpora cavernosa. In this picture, they're purple. And then the third one, remember, which includes the urethra, is called the corpus spongiosum. So in this picture, the corpus spongiosum is pink. Same thing over here. Well, remember we said that you could divide the penis up into the root, the shaft, and the glands. And then we've got these 3 chambers. So here are our 2 corpora cavernosa right there. And so the corpora cavernosa are dorsal. They're dorsal. And remember, they have this tunica albuginea around them. Well, if we go to the root of the penis right here, look what happens. They split apart like this. And those things sort of might look a little bit like legs. So they have legs, sort of. And the word for leg is crus, or crura is plural. So that's the crus of the penis right there, and that's the crus of the other one. Together it's crura. And those crura are attached to the pubic arch, to bone. They're attached with ligaments, and so they anchor the penis. If we look at this corpus spongiosum, look at it. On this end, at the root, it has a round structure. And that round structure is called the bulb of the penis. But if you follow it, look, it goes all the way to the end, and then it gets enlarged and round again. And the round structure on this end is the glans penis. So if you look at it here, you can see the 2 chambers. There's the corpora cavernosa and the corpus spongiosum. Corpora cavernosa don't go all the way to the end. They stop right here. They stop right here. But the corpus spongiosum does go all the way to the end and forms this glans penis. And then you get the bulb here and the crura here, again, are going to form the root. Well, if you look at it in cross-section, it's easy to see this tunica albuginea, and it's easy to see these vascular spaces in here that are going to fill up with blood. And so what happens when a male becomes sexually excited is this artery, which is right here in the middle, and right here in the middle, they dilate. So they open up, and so more blood flows to the area. And the capillaries in here are very, very, very leaky, which means that blood cells, blood and fluid leak out into the tissue spaces. And those tissue spaces, you can actually see them. That's what all these little spaces are in here. And so that fills up with blood. Again, there's these little tissue spaces. There they are as well. If we look at our real dissection, remember there's the testis, the epididymis, there's our ductus deferens, and then here is the penis right here. And so if we look at this, you can see the chambers. So this one and this one are those corpora cavernosa. This one is the corpus spongiosum. And remember, the corpora cavernosa have these sort of like legs. Again, they're called crura. And then there's the bulb of the penis. And there's the glands of the penis right there. Any questions? Well, we learned this already when we did urinary, but remember the bladder has the urethra coming from it, and the urethra runs through the penis. But it doesn't just run through the penis, it also runs through this gland, and it also runs through the abdominal wall. So if you look at the urethra, it has 3 sections to it. The part that runs through the prostate is called the prostatic urethra. The part that runs through the abdominal wall is called the membranous urethra. And the part that runs through the penis is either the penile or spongy urethra. Any questions? You can also see that the tissue changes. So the tissue in here, you should remember from A&P1, is transitional epithelium, and that runs through here as well. That's transitional epithelium. But when you get to here, the tissue changes and it becomes pseudostratified. And then when you get here at the very end, it changes again. It becomes stratified squamous. So 3 different types of tissue run through the penis and the reproductive system of the male. Any questions about the penis? Okay, well, we also have these accessory sex glands. So let's look at these. So accessory sex glands are the seminal vesicles, the prostate, and the bulbourethral gland. So let's look at the seminal vesicles first. So seminal vesicles are on the posterior side of the bladder. They're on the back side of the bladder, and there's 2 of them. And here they are right here. Well, they're glands. There they are right there. You can only see 1 in this picture. But if you look at them, they're definitely glands. And glands do secretions. And look, you can see all these little spaces in here where the secretion collects. Those are called secretory pockets, and they fill up with this fluid. The fluid is called seminal fluid. And this seminal fluid makes up about 65% of what comes out. So if we look at this seminal fluid, it's about 65% of what comes out. It's a viscous, which means it's thick fluid. It's alkaline, so it has a high pH. It's got stuff in it like vitamin C and stuff like that. But it's also got some kind of unique things in it. One unique thing it has in it is it has a lot of fructose in it. Fructose is the same sugar that you find in fruit. And that fructose, what it does is it allows the sperm to make ATP. So the sperm use it as an energy source to make ATP. So as the sperm come through here, then we're going to empty this fluid into the area where the sperm are. And now the sperm have lots of energy source to make ATP. Another thing that's found in the seminal vesicle is the same thing you find in blood, which is fibrinogen. Remember, fibrinogen turns into fibrin, and fibrin forms a clot. And so what happens when this fluid comes out is it very, very quickly coagulates. It clots. And then the last thing that's interesting about this is there's a bunch of hormones in here. These are eicosanoids, if you remember what we talked about with them. What's weird about them is they're called prostaglandins. Well, look at that word. It doesn't look like seminal vesicles. It looks like prostate gland. And that's because when these hormones were first discovered, that people thought they came from the prostate gland. And so that's what they named them. Well, the name has never been changed, but they don't come from the prostate gland. They come from the seminal vesicles. So there's our seminal vesicles. Again, they look like this if you look at them. Lots of these secretory cells, these little secretory pockets that fill up with fluid. And look, there's smooth muscle here. And that smooth muscle is going to contract and force its fluid into here. It's going to force it to come out. And what it comes out into is something called the ejaculatory duct. So, seminal vesicles. Any questions about them? Let's look at another one of these. This is called the prostate gland. Prostate gland is just single. There's not two of these. There's only one. The prostate gland is a little bit like a donut because it's got a hole in the middle. That hole in the middle is where the urethra is. So the urethra runs right through the center of the prostate gland. And what's going to happen to it when that fluid comes from the seminal vesicles, when the sperm comes from the ductus deferens, the prostate gland is also going to contract. There's smooth muscle around the outside of it, just like there was around the seminal vesicle, and it's going to contract, and its fluid is going to get forced into this tube. Well, when you look at it, this is what it looks like right here. Remember, the urethra also passes through here. And then there's the ejaculatory duct. We're going to force this fluid into this ejaculatory duct and into this urethra. And so it's going to mix with the fluid we already have. So if you look at this fluid, it's about 33%, about a third. And if you look at it, sort of white colored, it's milky colored. It has a slight low pH. It's slightly acidic. It's got things like vitamins in it and stuff like that. But the thing that's interesting about this is it has an enzyme in it. That enzyme is called fibrinolysin, and fibrinolysin breaks down fibrin. So remember, when this fluid comes out, it immediately coagulates, but it doesn't take very long for this fibrinolysin to chop up the fibrin, and then it becomes liquid again. So it becomes liquid. It starts out as liquid, and it becomes coagulated, and then it becomes liquid, all within a few minutes. If we look at the prostate gland under a microscope, you can see these secretory cells, tons of them, and tons of these secretory pockets as well. And again, the fluid is going to collect in these pockets. You can't see it in this picture, but outside around here is this smooth muscle, and it goes all the way around. And so what it's going to do when it contracts, it's going to force the fluid out, just like seminal vesicles. Any questions about the prostate? Okay, we have one more pair of glands, and these are little bitty glands. They're right here. Little bitty glands. They're called bulbourethral glands. They're called bulbo because they're on the bulb of the penis. And they're called urethral because they empty into the urethra. So, bulbourethral glands. They used to be called Cowper's glands. Cowper is a person's name. Remember, we're trying to get away from things being named after people. But they're not very big. They're about the size of an English pea. And these glands produce something called pre-ejaculatory fluid. Pre-ejaculatory fluid. In other words, it happens before ejaculation happens. Well, the reason why is because, remember, the urethra is a shared tube. Urine comes this way and comes out. And then the sperm and all those fluids come this way and come out. It's a shared tube. Well, urine is often acidic. And acid kills sperm. And so if there's any urine left in here, as the sperm came through, you'd start killing off the sperm. And you wouldn't want that to happen. And so what happens before ejaculation is these little glands release their secretion, and their secretion goes through the urethra and basically neutralizes it. So if there's any urine in there, it neutralizes it so it's not acidic. It also comes out the end, and if you come into contact with it, you would know that it's sticky. It's got mucus in it, or mucin in it. And that mucin is there to help with lubrication. So it acts like a natural lubrication. So it's this thick, clear fluid with a lot of mucus. Its job is to neutralize this acidity of the urine, and then it also aids in lubrication. Here's what they look like on the inside. And so look, you have tons of these mucus glands, a ton of this mucin in it. And there's our smooth muscle, just like before. So it's going to contract, and it's going to force this through the tube. And so here's a sort of a different view of all of it. This is a posterior view. So if you look at the posterior view, here comes our ductus deferens from the testis, and here comes the other ductus deferens from the testis. Here's our ampulla. That's where we're going to sort of sit and wait. And then there's the seminal vesicles. Again, there's two of these. Here's the prostate gland. There's our ejaculatory duct right there. And then here's the urethra, which comes down from the bladder. And then there's our bulbourethral glands right there. You can also, in this picture, see a nice view of the bulb of the penis and the crura of the penis, which, remember, forms the root. And in this picture, you can see the chambers. Here are the corpora cavernosa. Remember, there are two of those. And then here in the middle is the corpus spongiosum. Any questions? So that's where we're going to stop for today. Again, you can always email me with your questions, but we're going to stop. So thank you for coming, and I'll see you tomorrow if you show up tomorrow. Any questions?